Kristin, if you'd like to put your talk up and I'll start the introductions. So hello, everyone. Welcome to the first GCE for All of 2026. I'm John Lewick from now the snowy University of Rochester. My co-host today and always is Rick Cooley from the Just Getting Light Out Oregon State University. I'd also like to give a shout out to Kayla Jar-Weber, who is with us every week and keeps us on the straight and narrow. So this webinar series highlights the latest and greatest on genetic code expansion and all biology and chemistry that feeds into GCE. The GCE for All webinar happens every third Thursday of the month. All the webinars are recorded, so you can go back and watch them on the GCE for All website at your leisure. Like today, we have had some amazing talks, so please check them out. We often highlight principal investigators and leaders in industry in this forum, but also we'd like to highlight early career researchers, including graduate students and postdocs. So this is an excellent opportunity for postdocs looking to move on to faculty positions or industry positions to highlight their research. So if there's anyone that you'd like to highlight, please send us their information, and we'd like to give them the opportunity to speak during this webinar. Today, we're super fortunate. We have two fabulous speakers, Dr. Kristin Koutmou from the University of Michigan and Dr. Cheng Liu from the University of California, Irvine. So first up, we have Kristin. Kristin is joining us from University of Michigan, where she is an associate professor in the Department of Chemistry. Kristin received her PhD in chemistry from the University of Michigan under the mentorship of Carol Fierke, I think, where
Fierke, yep.
Where she had a series of really nice studies focused on the ins and outs of RNase-P and its interactions and processing of tRNAs. She went on to do a postdoc with Rachel Green at Johns Hopkins University, also HHMI investigator. She had beautiful studies on protein translation. Quickly then moved on to her independent career at University of Michigan in 2016 in the Department of Chemistry, and rose to the ranks of associate professor. She's also the co-director of the Chemistry Biology Interface Training Grant and the associate director of the Chemical Biology Program at the University of Michigan. Along the way, Kristin has received numerous awards and accolades for her research and also has garnered a large amount of funding from several sources to keep her research enterprise going. To name a few of her awards, we don't have time to name them all. She was awarded the Sihan Egi, maybe, Junior Faculty for Teaching and Mentoring in 2019, Dow Corning Early Career Professor of Chemistry in recognition of her research in 2023, and the RNA Society Award for Innovation in Research, Teaching, and Service in 2025. In short term, she's been extremely productive with publications and has already published 25 independent studies and incredibly has 8 that are submitted or in revision that I'm excited to read here in 2026. I'm not going to get into all of the impressive list of her current funding, but I wanted to highlight this one internal University of Michigan award, which is the Mid-Career Biosciences Faculty Achievement Recognition Award. And I mention this because I was recently told I was mid-career, and this made me reflect a little bit. I'm mostly muttering to myself, how the hell am I already a mid-career scientist? So, and I'd also like to say that I've been fortunate to collaborate with Kristin, and outside of being a fabulous scientist, I'd like to highlight the fact that I may be most impressed with the amount of energy and care that she puts into mentoring the people in her lab. So I'm excited to hear what Kristin has to say today, and today she'll be giving a talk on molecular level consequences of RNA modifications on protein synthesis. So Kristin, please take it away.
John, thank you so much. That is the most flattering introduction I've ever had. Thank you for that very sweet and kind and generous introduction. And I really also want to thank the community for inviting me to take part in this seminar series. Today, I'm going to be talking to you about a new story that's coming out of our lab, looking at the molecular level consequences of when the ribosome encounters inosine, which is, of course, one way that nature expands the genetic code. Sorry, technical difficulties. So the chemical diversity of RNAs far exceeds that of the 4 standard nucleosides, with over 150 different varieties of RNA modifications having been reported in non-coding RNAs, primarily in tRNAs and ribosomal RNAs. recent technology that has allowed us to map the location of modifications transcriptome-wide has also revealed that protein coding mRNAs can also be highly modified, with over 10,000 different sites of mRNA modification having been identified in eukaryotes. These modifications can really impact cellular protein production because they have the potential to modulate every step in the life cycle of an RNA after it's been made. It can change structure and dynamics, splicing and maturation, the stability or half-life of an RNA, how it interacts with other biomolecules in the cells like proteins and RNAs, in order to ultimately influence the function and ability of the RNA to do its job properly. This is particularly true in the context of translation, where the ribosome decoding center really relies on reading discrete hydrogen bonding interactions between an mRNA codon and an incoming tRNA anticodon in order to ensure that translation is rapid and accurate and that the genetic code is properly decoded. You can imagine that any small perturbation to these hydrogen bonding interactions can really have an outsized effect on the accuracy of translation decoding. And this is important not only for biology, but also in the context of many ribosome-dependent therapies that are emerging. For example, mRNA vaccines and therapeutics, mRNA editing, which actually relies entirely on inserting an inosine, the base we're talking about today, into an mRNA, and through possibly modulating the ability of tRNA suppressors, which are tRNAs designed to read through stop codons and treat disease caused by premature termination. However, I would like to argue that we should not be putting the cart before the horse as we are developing these RNA technologies, which clearly have a lot of promise. So there's a lot of work going into engineering different RNA sequences and different varieties of therapeutic modalities. However, we don't still understand really on the molecular level how these modifications are impacting the translation systems and exerting their effects on these therapies. Some examples of modifications we know are important or might be important for these different therapies are shown here. And really the underlying theme in my lab is we're trying to understand how modifications impact translation and protein production. And some of our initial work, and I would say the work we're best known for, has really focused on looking at the modifications pseudouridine and M1 pseudouridine, which are found in all of the mRNA vaccine and therapeutic platforms that are currently available. Our lab has discovered that pseudouridine changes the speed and accuracy of the ribosome and can actually promote a little bit of recoding. We're also interested in pseudouridine in the context of mRNA editing because we've demonstrated that pseudouridine can promote recoding, as well as inosine in the context of RNA editing, which is sort of the classic way to edit RNAs in vivo. And that's going to be the story I tell you about today. Additionally, we've worked quite a bit on trying to understand how modifications in tRNA impact the correct tRNA insertion, with an idea of trying to think about which modifications we might want to be incorporating into these tRNA suppressors in order to promote their ability to work. The focus of my talk today is going to be on inosine. So why inosine? Inosine is incorporated in eukaryotic systems through a system called A to I editing. During A to I editing, there's an adenosine molecule that is modified by an enzyme called an ADAR. ADAR is an adenosine deaminase that converts adenosine into inosine, replacing the amine with a carboxyl group. A to I editing has been known for a long time to make some really key contributions to biology. It's an important marker of self in the immune system. It can modulate splicing, and importantly, for the purposes of this particular interest group, it can also result in the recoding of mRNAs. That's because inosine can base pair with cytidine, which essentially recodes an A into a G in the context of the ribosome, which is relying on discrete base pairing interactions to choose which tRNA comes in. And shown here is a GC base pair versus an IC base pair. And you can see that the orientation of the bases is quite similar. It looks more or less like a canonical Watson-Crick base. However, it's, of course, lacking one of the three hydrogen bonds. There has been an incredible amount of effort put into developing ways to selectively add inosine to mRNAs in order to suppress nonsense mutations, which actually account for a very large fraction of all known genetic inherited diseases. The idea behind this is to convert a stop codon, like a UGA codon, into a sense codon, in this case, a tryptophan codon, simply by editing that A to an I, where now it's no longer read as a UGA stop, but as a UGG tryptophan codon. And there's been a large amount of success in this field, and there are a number of companies that are working to figure out ways to selectively send these ADAR enzymes to specific sites and fix genetic mutations. However, much to my surprise, as somebody who's been working on modifications for a while, when I started to try to dig into understanding how inosine is exerting its effect, in order to make all of these companies and their technologies possible, it became readily apparent that no one had ever looked at how inosine impacts the speed or accuracy of the ribosome on a molecular level. And that knowledge gap is really what the work that I'm talking about today is hoping to try and fill. So this is actually a very difficult question to ask in the context of a cell, to look at the speed of a ribosome on an individual codon, or to look at how it gets recoded, to what extent, what it's getting recoded with, and so on. So in order to get around this problem, my lab turned to a tool that we have present in lab, which is a fully reconstituted translation assay. In this assay, we purify out all of the components necessary for translation. We reassemble them, and we can watch the formation of individual peptide bonds. We started off by asking a really low-hanging fruit question, which is what happens when inosine is present in all three positions of a codon that's poised in the empty A site of a ribosome, ready for an amino acid to come in and react with it. And this work was spearheaded by a now fifth-year graduate student in lab, Rachel Giles. In these assays, after we mix all of our components together, we quench the assays at discrete time points on the millisecond time scale and watch the formation of a dipeptide bond. This allows us to do transient kinetics with the ribosome. So when we do this, we see that inosine slows amino acid addition by cognate tRNAs in a context-dependent manner. So what this means is we are reacting our inosine-containing codons with a tRNA that reads inosine as a G. In this case, we can see that having an inosine in the first position slows the ribosome substantially by about 8-fold, but having it in subsequent second and third positions doesn't make a large impact on the ribosome. We also compared this to having an unedited transcript around, where now instead of having an I, we have an A. And we see that it reacts more robustly than having the unedited version of the codon, which is what you would expect. Something I haven't told you about is that while inosine does recode by base pairing with cytidine, inosine is unique because it base pairs not only with cytidine, but also with uridine and adenosine, really giving it an expanded ability to perhaps select unanticipated RNAs in the cell. And we know that this can happen in vivo by some beautiful reporter work done by the group of Michael Jansch in Austria, where he made a reporter that he expressed in rabbit reticulolysate, where he had an inosine, a single inosine in this reporter sequence. And he then looked by mass spectrometry to see what amino acids got put in depending on where the inosine was within the codon. And I don't need you to fully understand this plot. The take-home message from this slide is really that he didn't only see one thing get produced. He saw inosine get decoded as a G, as shown here with all of these blue spots, but he also saw it in vivo, or in lysates, I should say, get recoded as a G, an A, or a U, suggesting that there's more than just this ability to be read as a G playing a role in what protein is ultimately going to get made when an inosine is present in the codon. So we wanted to see if we could recapitulate this or gain some understanding as to why this might be happening, if it's really possible, a fundamental principle that's being driven by inosine base pairing. So in order to do that, we looked at interactions where we had inosine base pairing with either a G, which doesn't alter ribosome kinetics. We had it base pairing with an A, where now it's being decoded by a U, and we saw that yes, this actually increases amino acid addition, suggesting that this could be a reasonable target for something to get added in the cell. And the same thing when it's base pairing with a U and being decoded now as an A. So this demonstrates that there's more than one way to recode with inosine in an mRNA codon. All right, so everything I've shown you thus far is pretty clear. It's obvious that inosine behaves like we imagined it would, right? Everything that we have from in vivo studies suggested that inosine should primarily be read like a G by the ribosome, but sometimes it can add other things. None of that is particularly, and that it slows the ribosome down. So none of that is particularly earth-shattering, but it is useful information. The next things I'm going to tell you are brand new. They add things to the field that didn't exist before. It turns out that we had no crystal structures of the ribosome on an inosine containing codon, despite thousands of studies looking at inosine in cells, primarily, again, in the context of trying to drive recoding by ADAR enzymes. So in order to ask what this might look like and what on a molecular level might be driving these changes in recoding and ribosome speed that we've seen in the test tube, we worked with Professor Kristin Koutmou at Emory and her student Chipo in order to get the first cryo-EM structures of a ribosome sitting on an IUG codon bound to a tRNA valine, where it should be, which should decode the inosine as a G. And to our surprise, when we did this, the tRNA did not look exactly like it does on a GUG codon. While we did see that the ribosome maintained its Watson-Crick geometry when there was a modification in the first site, in the first position of the codon, we saw that the GU at the wobble pair, which is shown right here, is actually substantially elongated by having an inosine 2 positions over. I mean, it's elongated from a normal hydrogen bonding distance of 2.9 angstroms out to 4.1, which is really almost outside of hydrogen bonding distance. We also saw that C36 in the tRNA is no longer planar with inosine, like it is during a normal, fully accommodated conformation. So this structure told us that, our kinetics makes sense. The tRNA isn't coming in quite like you would expect it to if this was fully a normal cognate interaction. But this is just a snapshot of what is happening in the ribosome. This is, of course, a very dynamic process. So we next wanted to ask, can we get some more molecular logic as to what it is we're going on in, or these observations we've made in our kinetic platforms, as well as in the EM structure? To do this, we worked with Professor Stacy Wetmar and her student Mark at University of Lethbridge to develop some really advanced MD simulations for looking at these codon-anticodon interactions. They modeled the entire A site of the ribosome with a tRNA bound to EFTU present. They did a 1 microsecond all atom simulation, which if you do simulations, you'll realize is a huge simulation, in fact, in order to try to get some molecular logic for the observations we were making. And when they did this, they saw that the cognate tRNA really samples unproductive conformations much more frequently when an inosine is around. They noticed that some of the key residues that are important for monitoring codon-anticodon interactions, A1492 and 1493 and G530 in the ribosome, are disengaged far more often when an inosine is present, right? We're talking if they're engaged 45% of the time when an inosine is present, as opposed to 93% of the time when an unmodified codon is there. There's also premature tRNA straightening, which means that the tRNA isn't being correctly accommodated. And you can really see that in these videos of their simulations, which are not playing. Let's see if I change it. Oh, there we go. So here in the first one, you can see that this is a simulation of the normal codon. Where if you look here, you'll see it's shaking around, but it's pretty well accommodated. There's not a lot of swinging of these 1492, 1493, or G530 bases in and out. This is going to look different in the simulation here shown on the right with the inosine codon. Where now, if you watch closely, you're going to see disruption between that tRNA codon-anticodon and the surrounding ribosomal sites, where this guy's really moving around quite a bit. And this is, again, totally consistent with our kinetics that show that the ribosome adds amino acids much more slowly when there's an inosine in the first position of a codon. So the MD is predicting a context-dependent tRNA veiling accommodation, where it's more accepted when there's inosine in the second and third position, consistent with our kinetics. And similar computation looking at near cognate codons, where now we're reading inosine as an A, demonstrates the opposite trend, where now inosine is more likely to be in a favorable conformation than the non-cognate or near cognate GUG codon. So together, this gives us a model for what's happening when the ribosome encounters an inosine in a codon. So in the context of a cognate codon where I is being decoded as a G, the mini helix, meaning the anticodon stem loop helix, is misaligned. The key rRNA bases, A1492, 1493, and 530, are disengaged. The tRNAs in a straight state, in a rejected state, and the ribosome is slowed. However, when there is an inosine at the second or third position, now we see this mini helix is properly aligned. We see those key rRNA bases are engaged. The tRNA is bent like it's supposed to be, and it's an accepted preferred state. On a near cognate codon, when we see that this, while amino acid addition is generally slow, when there's an inosine present, this slowness is sped up as the mini helix is realigned. Those ribosomal side chains are re-engaged, and you get an enhancement in the ability of the ribosome to add near cognate tRNAs. So in sum, I hope that I've convinced you that there is something interesting going on the molecular level when the ribosome is dealing with inosine. It's not just solely read like a guanosine, and is going to always reproducibly give you the exact same thing. And there really are some fundamental differences with how the ribosome views that codon. I'd like to thank my fantastic lab, the awesome students and collaborators, in particular, highlighted here in red, and my funding sources, and I'm happy to take any questions.
That was great, Kristin. I'm gonna start off with some questions. Well, people can either put it in the chat, and then I can read them too, or we can allow you to ask the questions yourself. So start off well, that was really fantastic. So there's all the rules for recoding mRNAs for fast translation likely don't incorporate anything to have to do with inosine. Is that true, or is that not true? Or do you think
What do you mean for fast translation?
So what people usually do is they codon optimize to increase the expression. And so
Yep.
I'm guessing that this is not incorporated into those algorithms because people don't understand it yet.
That would be my guess, yeah. Not to my knowledge.
And so it's likely that you can tinker with translation and production of protein much more if you take these into account.
100%. Yeah. And I mean, this is particularly true in the context of tRNAs, right?
Yeah.
Where you absolutely, I would argue, have to take tRNA pools and what modifications are on the tRNA and into account when you're thinking about codon optimizing an entire gene.
Yeah. And then when you showed your videos, you showed that the anticodon is coming off of the codon and flipping back on and off. Is that a water-filled vestibule? So is it exchanging interactions with water in the A site? Is that what's happening?
Yeah.
And can you modify that in any way? So is that modifiable with changing the environment, or is it kind of?
So I think that's a hard question to ask, because how are you going to exclude water from the A site of the ribosome in a practice? You're not going to be doing these reactions in DMSO.
Right. Okay. Are there any other questions from anybody? Andy?
There's one in the Q&A.
Oh, there? Okay.
And I have a question too after that.
You can go ahead, Andy. I'll read this.
Oh, just a pretty quick. There was the round figure you showed that showed in some places it was only used as a encoded one way, and in other places it was a red dot because you could put 3 different amino acids.
Yep.
Does that mean, anyway, what's the difference between those from your understanding? Because in some cases, you'll get a pure form of a protein. And in the other case, you'll get a protein with 3 different amino acids all in the same place, and it's a mess. So if inosine is going to be used, it seems like you only want it to be used when it's going to give a single amino acid translation.
Yeah, right. So this is the argument we're making both with inosine, and we've made this argument a lot with pseudouridine, which I chose not to talk about today. But yeah, so that's the $1 million question. It's also extremely context dependent, depending on where things are within the codon, whether or not you're going to be able to make one thing or 3 things. And that's my take-home message would be, if I was designing these things as therapeutics, I would surely be doing some really robust mass spectrometry on my proteins that I get out to make sure I'm getting more than, only the thing it is that I expect that I'm getting. I can tell you for sure, with pseudouridine, when we've done this in the context of completely pseudouridinated mRNAs, we get all sorts of, they're very low levels, but you get low levels of all sorts of things.
Okay, thank you. Caution.
Yeah,
Yeah.
caution. I mean, sometimes it could be good. Like if you want to make low levels of products for eliciting an immune response for a vaccine, that might not be horrible to give yourself a variety of peptides to display. But if you want to do protein replacement therapy, it's a different deal.
Yeah. All right. So the question online is, well, first, great talk and beautiful complete story in these post-transcriptional modifications. Can you comment on which endogenous genes is the A to I more prevalent, or is there some sort of physiological condition or stress conditions where this A to I conversion would be beneficial? And then one more, or what percentage of the transcriptome contains A to I modifications?
So this is an extremely species dependent answer. So there are a lot of genes, particularly in immune genes, where A to I editing is quite present. There are some physiological conditions where the conversion is beneficial. I think there's some really great work by Josh Rosenthal's lab that shows this in particular in octopus. There's also some nice work out of Peter Beal and Sheila David's lab that shows this in human cells where it's where there's a recoding of a DNA damage pathway that alters the activity of that DNA damage enzyme. So there's lots of kind of one-off examples of these things, but there's not a really holistic view on where all the recoding is happening. It's also tricky because it's not just recoding in terms of translation when you're looking at the biological function of these A to I sites, but most A to I sites are actually in UTRs and are really probably screwing with splicing.
That's great. I mean,
Very
if anyone's
cool.
interested in A to I, like just in want to do kind of fun reading, the stuff in Octopus is like really
Oh,
fun.
it's so cool. Yeah, it's amazing. It's a major mechanism in cephalopods, period.
Yeah, super
Very
cool.
cool.